Thank you for clicking on the link for this introductory lecture on PTEN deletions and clinical outcome in prostate cancer. So this first part, I'm going to give some background to PTEN and also describe some earlier studies that we did looking at PTEN by FISH in PIN cells. So I'm speaking to you from Queen's University in Kingston, Canada, and thank you. Let's get going. So the consequences of earlier detection of prostate cancer by the public health screening systems such as PSA screening, digital rectal examination, means that the incidence of prostate cancer has actually increased. And something like 90% of men now diagnosed with prostate cancer have much earlier clinical presentations, so smaller tumors, more localized disease. However, some of the unfortunate thing is that about 20% of these earlier detected tumors, if you like, actually are ones that have more aggressive behavior and a high potential for recurrence or metastasis. So really the challenge that my lab has been involved in has been to develop markers that can help identify the tumors that are likely to have this more aggressive phenotype. This is a typical presentation after radical prostatectomy of these sort of earlier detected tumors. So you can see an area on the top right of this slide, which is a very small region of prostate cancer. Also, on this area, there are some regions that are called PIN, which I'll come to in a minute. And so taking a closer look at this tumor, it's a Gleason 7 tumor, and fortunately this man it was detected before it spread outside of the capsule. So this is one of the consequences and the benefits, I think, of the detection screening programs. So let's just say a little bit about PTEN, because that's really been a central part of our work over the last 5 years. This, of course, is a tumor suppressor gene. It's a phosphatase, and the reason it's called PTEN is phosphatase and tensin homolog. And in fact, the gene itself is present on chromosome 10, so it's fairly easy to remember. It's involved in regulation of the cell cycle by preventing cells from growing and dividing too rapidly. And it was identified as a tumor suppressor gene by virtue of the fact it's inactivated, and this is usually by deletion of the entire gene in prostate cancer. In other tumors and in other patient classes, it's actually seen by point mutation, but in prostate cancer it seems to be mostly by deletion of the entire gene. So how does the gene itself function? Well, when the gene is doing its job, the protein that's produced signals cells to stop dividing, and in fact causes them to enter a programmed cell death pathway when adequate growth has taken place for the normal function of the cell. In the case of prostate, this would be to populate the gland and provide its secretory function. So loss of these critical regulatory functions by loss of the gene, of course, removes this restriction of cell growth, so that this uncontrolled growth then leads to the formation of tumors and prostate cancer in the case of cells arising in the prostate gland with these defects. There's also evidence that the protein is involved to a number of different functions, such as cell movement, migration, adhesion, and these are properties that we know are related to more aggressive behavior, such as metastasis. In addition, the protein has some role in stabilizing DNA, so loss of this essential protein clearly creates an enormous challenge to cells that bear such a loss. Now, this is a slide from our first paper really looking at PTEN, and it was published here, Cancer Genetics and Cytogenetics, some 5 years ago. This is an area that contains this cell types which I call PIN, which stands for prostatic intraepithelial neoplasia, and this is considered a precursor to prostate cancer. So in our first use of the assay, we did a 2-color FISH assay, a commercial probe, and we found that in some of the cancers in this study we saw normal PTEN. Other cancers had lost 1 copy of the gene, so instead of having 2 red spots, they have just 1 red spot. You can see that here in comparison to this one. And then, interestingly, some of these high-grade pins also had lost 1 copy of the gene, and you can see high-grade pin here and here that just has one copy of the gene. And in fact, when we looked at our whole series, we didn't see any losses of PTEN in benign glandular epithelium, or in a sort of related precursor called low-grade PIN that isn't much studied now. But in high-grade PIN, 3 of the 13 PIN lesions we looked at had a deletion of PTEN, and in this particular cohort, which was quite an aggressive series, 22 out of 35 had lost one copy of the gene, and 2 out of 35 had lost both copies of the gene. So this was our first study using PTEN, looking at prostate cancer. And in the subsequent slide series that you can download and look at, I'll be describing more about PTEN FISH assays in cancer. So thank you for listening to this first part.